Welcome back. Today our topic is molecular basis of inheritance. In principles of inheritance, we have studied that how chromosomes, how inheritance will pass and how the genes are passing from parents to offsprings. But in this lesson, we are going to study what is the basis, what is the basic things for inheritance, how the inheritance will pass from parents to offsprings, and how inheritance is a mechanism of inheritance we are going to see in this lesson. So now Mendel used the word factors. Mendel used the word factors for genes. We know that genes are responsible for inheritance. Genes are responsible for inheritance. But Mendel did not use the gene. Mendel did not use the word gene. The word gene is proposed by Johansen. The word gene is proposed by Johansen. And the term chromosome was proposed by chromosome. The term chromosome was proposed by Waldeyer. The term gene was not used by Mendel. Mendel used, instead of genes, he has used the word factors. Genes, the term gene is given by Johansen scientist. And chromosome, the term chromosome is given by Waldeyer. And Mendel used the factors. His factors only responsible for inheritance. The factors only, Mendel factors only responsible for inheritance. We have studied in principles of inheritance lesson. And so genes are present in DNA. Genes are present in DNA. In fact, gene is nothing but a segment of DNA. Gene is nothing but a segment of DNA which is responsible for production of a particular protein, or which is responsible for carrying genetic information. Gene is nothing but a segment of DNA which contains genetic information, or which contains the information for coding a protein. Gene is nothing but a segment of DNA which contains the information or genetic information for preparation of proteins, or gene is a segment of DNA which contains the information for which contains the genetic information. So this is gene. Next, where the genetic information, where the DNA is present? DNA is present in nucleus. DNA is present in nucleus. Inside the nucleus, a genetic material is present, that is DNA. So genetic material is present inside the structures called, rod-like structures called chromosomes. Inside the nucleus, DNA is present in rod-shaped structures called chromosomes. The chromosomes made up of DNA and some proteins, DNA plus some proteins called histone proteins. Inside the nucleus, chromosomes are the rod-like structures. The rod-like structures are called chromosomes. The chromosomes are made up of DNA and some proteins. So these proteins are called, the proteins associated with DNA are called histone proteins. These proteins are called histone proteins. Histone proteins along with DNA forms nucleosomes. The nucleosomes form chromosome, and chromosome only present inside the nucleus. Okay. Now let us see the structure of DNA. DNA is also called as a polynucleotide strand. Polynucleotide strand. DNA is a polynucleotide strand. And what are the monomers? The monomers are nucleotides. The monomers are nucleotides. DNA is nothing but a polynucleotide strand, and monomers are the nucleotides. Means many nucleotides. Nucleotides are the units of DNA or polynucleotide strand. Or polymers, DNA is a polymer and nucleotide is a monomer. DNA is a polymer of nucleotides. DNA is a polymer of nucleotides, and nucleotide is the monomer of polynucleotide strand. And each nucleotide is made up of 3.3. A nucleotides. For example, if you take in human beings, how many nucleotides, human DNA. In human DNA, how many nucleotides are present? Human DNA consists of 6.6 into 10 to the power of 9 nucleotides. And E. coli contains 4.6 into 10 to the power of 9. And lambda phase contains 48502. 48,502 nucleotides are present in its DNA, and 5x174 bacteria. 5x174 bacteria contains 5386 number of 5,386 nucleotides present in the DNA. So nucleotides are the monomers of polynucleotide strand. Nucleotides are the monomers of polynucleotide strand. Human DNA is made up of 6.6 into 10 to the power of 9. So as we know, DNA, your total DNA is nothing but total DNA, genome is half of the DNA. Genome is half of the DNA. It consists of 3.3 into 10 to the power of 9. Total human DNA, if you take 6.6 into 10 to the power of 9, and genome is half of the DNA or half of the chromosome number of any organism is called genome. So that human genome consists of 3.3 into 10 to the power of 9 number of nucleotides. Human DNA consists of 6.6 into 10 to the power of 9. Genome is half of the DNA or half of the chromosomes, so that it is 6.6 into 10 to the power of 9. And next, let us see E. coli. E. coli DNA is made up of 4.6 into 10 to the power of 9 number of nucleotides. And lambda phase is made up of, lambda phase DNA is made up of 48,502 number of nucleotides are present in its DNA. And next, 5x174 bacterium DNA is made up of 5,386 number of nucleotides present in its DNA. Now, structure of polynucleotide strand. DNA is a polymer of nucleotide, so that the monomers of DNA are nucleotides. So each nucleotide contains three things. The first one is pentose sugar. The first one is pentose sugar, and the second one is nitrogen bases, and third one is phosphate group or phosphate moiety. There are three components of nucleotides. The first one is pentose sugar, and the second one is nitrogen bases, and the third one is phosphate group. Now let us see the pentose sugar. Pentose sugar also called ribose sugar. Pentose sugar is made up of C5H10 and O5. Pentose sugar is made up of 5 carbons and 10 hydrogens and 5 oxygens. So the formula of ribose sugar is C5H10O5. So this is the structure of ribose sugar. So ribose sugar is made up of totally 4 free OH groups. This is one OH group, and this is hydrogen, and this is one OH group. So totally this is the first carbon OH group, and this is second carbon OH group, and this is third carbon OH group, and this is fifth carbon OH group. So a pentose sugar contains totally 4 free OH groups. There are totally 4 free OH groups. So this is the first carbon OH group, and this is the second carbon OH group, and this is the third carbon OH group, and this is fifth carbon OH group. So totally carbons, how many carbons are there? 1, 2, 3, 4, 5 carbons. CH2O. This is fifth carbon. So this is first carbon, this is second carbon, this is third carbon, this is fourth carbon, and this is fifth carbon. At the fifth carbon, this is one OH group is present. So totally 1, 2, 3, 4 free OH groups are present in a ribose sugar. A ribose sugar combines with ribose sugar plus nitrogen base. A ribose sugar with nitrogen base is equals to nucleoside. A ribose sugar plus nitrogen base equals to nucleoside. And a nucleoside equals to a nucleoside plus and phosphate group equals to nucleoside, and phosphate group equals to nucleotide. So this is totally the structure of a nucleotide. So nucleotide consists of three things. One is ribose sugar, another one is nitrogen base, and another one is phosphate group. Ribose sugar plus nitrogen base together called ribose sugar and nitrogen base together called nucleoside. And nucleoside plus phosphate is called nucleotide. Now let us see nitrogen bases. Nitrogen bases are two types. First one is purines and second one is pyrimidines. Purines and pyrimidines. Purines and pyrimidines. First one, let us see purines. So this is the structure of adenine. Two heterocyclic rings are present in purines. Only one heterocyclic ring is present in pyrimidine. Two heterocyclic rings are present here. This is adenine. And the next one is thymine. Thymine contains and this is guanine, and next and this is cytosine, and next and this is thymine, and this is uracil. So these are totally nitrogen bases. Their nitrogen bases are two types, purines and pyrimidines. So first one is adenine. So this is adenine. So adenine is made up of totally two heterocyclic rings. And next one, this is guanine. So this is the structure of guanine. So guanine is also made up of two heterocyclic rings, so that these two are called purines. Adenine and guanine, these two are purines. Next, cytosine. Cytosine is so made up of only one heterocyclic ring, and this is thymine. So cytosine and thymine. So now let us see thymine and uracil. Thymine and uracil both are having the same type of structure, but here one difference is there, that is in the case of thymine, this is CH3 is present in the fifth carbon. This is 1, 2, 3, 4, 5. In the fifth carbon, methyl group is present. In the fifth carbon, methyl group is present. So the thymine is called 5-methyl uracil. 5-methyl uracil. So that in thymine, uracil is modified into thymine. Thymine is also called as 5-methyl uracil. In the thymine and uracil, if you see, thymine and uracil, so the structures will be looking similar. thymine and uracil structures, but only one difference, that is in the uracil, there is the fifth place, there will be no methyl group here in this, there is no methyl group in the fifth carbon, but in the thymine there is a methyl group in the fifth carbon. So that thymine can be considered as 5-methyl uracil, because uracil is not a stable compound, uracil is not a stable pyrimidine, so that uracil is modified into 5-methyl uracil, so that 5-methyl uracil only thymidine. A purine is always paired with pyrimidine. Purine is always paired with pyrimidine. A purine is always paired with pyrimidine. For example, purine, if you take A, adenine always paired with thymine with 2 hydrogen bonds, and guanine is always paired with 3 hydrogen bonds with cytosine. Adenine always paired with thymine, and guanosine always paired with cytosine. And what about uracil? Where uracil is present? Uracil is present only in RNA, and thymine is present only in DNA. So if you take in the case of DNA, adenine is paired with thymine with 2 hydrogen bonds. In the case of RNA, adenine is paired with uracil. So in the case of RNA, thymine is replaced by uracil. In the case of DNA, uracil is replaced by thymine, because uracil was the less stable pyrimidine. Uracil is the less stable pyrimidine, so that it was present in RNA only. In the later stages in the evolutionary order, what happened? The uracil, it was very less stable pyrimidine, so that uracil was replaced by thymine in DNA. So in the DNA, thymine is present, so that it is somewhat more stability. It got somewhat more stability. And cytosine is present in commonly present in both DNA and RNA. Cytosine is commonly present in both DNA and RNA. So that cytosine can be considered as the common pyrimidine. Cytosine can be considered as the common pyrimidine in both DNA and RNA. Cytosine is the common pyrimidine for both DNA and RNA. So that is, purines and pyrimidines, in this way they will always get paired with each other. Nucleoside equals to ribose sugar plus nitrogen base. A nitrogen base connected to ribose sugar and forms a nucleoside. So nucleoside equals to ribose sugar plus nitrogen base. So this is one ribose sugar. In one ribose sugar, the formula of ribose sugar is C5H10 and O5. So there are totally how many free OH groups are there? There are 4 free OH groups are present in a ribose sugar. In a ribose sugar, there are totally 4 free OH groups are present. So this is first carbon OH group, and this is second carbon OH group, and this is third carbon OH group, and this is fifth carbon OH group. There are totally 4 free OH groups are present in a ribose sugar. In this ribose sugar, when this ribose sugar is paired with the nitrogen base, in the nitrogen base, this OH group is nitrogen base replaces the OH group present at first carbon. Second carbon OH group will be left like this only, and third carbon OH group this is, and this is fifth carbon, and this is fifth carbon OH group. So now this is now called as, because this is one ribose sugar, and but here there is no base, but this ribose sugar contains base here now. So the base replaces this OH group. This OH group is replaced by this base, and this OH group, from the base one hydrogen will be replaced in the form of water. In the nitrogen base one hydrogen and this OH group both together removed in the form of a water molecule. So that this bond is called glycosidic bond. This bond is called glycosidic bond. The glycosidic bond is formed in between nitrogen base and ribose sugar. When the ribose sugar combines with the nitrogen base, a glycosidic bond is present. In the glycosidic bond, one water molecule is removed, and that water molecule is formed by the one hydrogen present in the base and this OH group. So that this OH group is replaced by base. And phosphate group next. Phosphate group, now this is first carbon, this is second carbon, this is third carbon, this is fourth carbon, this is fifth carbon. Now phosphate group replaces the OH group present at third carbon. Third carbon OH group is replaced by one phosphate group, so that this is called 3' end, and another phosphate molecule replaces OH group present at fifth carbon, so that this is called 5' end. So this is 5' end and this is 3' end. So what is 3' end? That phosphate replaces one OH group present at third carbon, so that this is called 3' end, and phosphate replaces one OH group present at fifth carbon, so that this is called 5' end. Chargaff rule, it was proposed by Erwin Chargaff and it was popularized as Chargaff rule. And Chargaff rule is the ratio of purines and pyrimidines is equals to, the ratio of purines and pyrimidines is equals to 1. So that is A plus G purines and T plus C equals to 1. This is Chargaff rule. Adenine plus guanine by thymine plus cytosine equals to 1. So this is adenine guanine. The meaning of this Chargaff rule is adenine guanosine together how much? That will be equal to thymine and cytosine. The sum total of adenine plus guanine is equals to the sum total of thymine plus cytosine. The sum total of adenine plus guanine is equals to the sum total of thymine plus cytosine. Let us explain it very clearly. For example, here one question is there. If a DNA contains adenines 30%, then what is the percentage of cytosine? That is the question. If a DNA contains 30% of adenines, then what is the percentage of cytosine? Now let us take that A plus G and T plus C Chargaff rule equals to 1, right? So let us substitute this 30 instead of A. Adenine is 30%. Plus adenine is 30% means adenine is always paired with thymine, right? Adenine is always paired with thymine. So the thymine also obviously 30 will be there, no doubt. Adenine is always paired with 13. If the polynucleotide stand, adenine is one side is there means thymine is another side. So that adenine and thymine always paired with 2 hydrogen bonds we know. So there is no other alternate in DNA. In DNA, adenine always paired with thymine. So that the adenine percentage is 30 means thymine percentage will be 30. Adenine percentage will be 30 means thymine percentage also will be 30. So that remaining 30 plus 30, how much percentage is over? 60% is over. And remaining how much is there? 40% is there. In the 40%, G plus C equals to 40. G plus C equals to 40 means G equals to 20 and C equals to 20. C equals to 20. Then 30, 30, 20, 20 equals to 1. This is ratio of purines and pyrimidines in a DNA, and this number is always equals to the sum total of purines and is equals to the sum total of purines is equals to the sum total of pyrimidines. So this is Chargaff rule. A plus G by T plus C equals to 1. So this is Chargaff rule. Based on the Chargaff rule, if one nitrogen base percentage is given, then the remaining all nitrogen bases we can find out. Here let us see what is the main difference between DNA and RNA. This is ribose sugar. In the ribose sugar, the first place, this is first carbon OH group and this is second carbon, this one OH group and this is 2 OH group and this is 3 OH group and this is fifth OH group. So this is the OH group present at first carbon, this is second carbon, this is third carbon, this is fifth carbon OH group. The first OH carbon is replaced by nitrogen base. The OH group of first carbon is replaced by nitrogen base, and the second OH group is replaced by third OH group is replaced by one phosphate group, so that this is called 3' end, and fifth OH group is replaced by another phosphate, so that this is called 5' end. So base replaces first carbon OH group and third carbon OH group is replaced by one phosphate, and fifth carbon OH group is replaced by another phosphate. So remaining one OH group is left. Now this is called ribonucleotide. So this ribonucleotide is present in, this is ribonucleotide. So ribonucleotide means 2' OH group, 2' OH group is present in ribonucleotide. So in the ribonucleotide, 2' OH group is free, so that it is highly reactive, so that RNA is having less stability because of presence of this OH group. This OH group is highly reactive, so due to presence of this OH group, second OH group, the ribonucleotide or RNA is highly less stable. So this is 2 OH group. So 2 OH group is present in ribonucleotide. So same type of nucleotides are present in all nucleotides are having second OH group in RNA. Whereas in DNA, this OH group is replaced by one hydrogen here, so that the oxygen is removed, so that this is called deoxyribonucleic acid. Deoxyribonucleic, because here OH group is not present, OH group is replaced by only one hydrogen. In the case of DNA, in the 2' OH group is absent. 2' OH group is absent. If 2' OH group is absent, that is called DNA, deoxyribonucleotide. If 2' OH group is present, then this is called only ribonucleotide. If it is OH, then that is ribonucleotide. If it is only H, then this is DNA, deoxyribonucleotide. So that is the main difference between DNA and RNA. In RNA, all the nucleotide contains OH group in the second place, so that this is called 2' OH group. So 2' OH group is present in all the nucleotides of RNA, and 2' OH group is absent. Here oxygen is absent, so the deoxy. In the ribonucleotide, one oxygen is removed, so that this is called deoxyribonucleotide. In somewhat clearly what we can say is 2' deoxyribonucleotide. The DNA is called 2' deoxyribonucleotide, because the deoxy means oxygen is removed. In which place oxygen is removed? In the second carbon place. This is second carbon place. In the place of second carbon, oxygen is removed, so that this is called deoxyribonucleotide. 2' deoxyribonucleotide. All this type of nucleotides are present in DNA. In the case of RNA, all the nucleotides contains OH group in the second place. So that this is the main difference between DNA and RNA. Due to presence of this OH group at the second place, RNA is less stable than that of DNA. This is polynucleotide strand. The polynucleotide strand consists of 2 strands which are passed in 5' to 3'. This is 5' to 3' direction, and this is 3' to 5' direction. You can see this is a polynucleotide strand, and this is which group? This bond is called phosphodiester bond. This bond is called phosphodiester. In between two nucleotides, a bond is present called phosphodiester bond. And this is one nucleotide, and this is one nucleotide, this is one nucleotide. A nucleotide contains a base and phosphate groups. So this is, as it forms, phosphate group binds to ribose sugar with ester bond. Both sides it is with ester bond. 2 phosphate groups are bind to a nucleotide, so that this is called phosphodiester bond. In a polynucleotide strand, 2 nucleotides are linked together by phosphodiester bond. In a polynucleotide strand, 2 nucleotides are binded together with phosphodiester bond, and this is base, and in between the bases hydrogen bonds are present. These are hydrogen bonds, and this is one strand and this is one strand. So this is 5th carbon, and this is 5 prime 5 prime, this is 5 prime end, and this is 3rd carbon, and this is 3 prime end. So this strand is passing in this direction, 5 prime to 3 prime, and this is passed in this direction, that is 3 prime to, this is passed in this direction, that is 3 prime to 5 prime. So 3 prime to 5 prime. So totally 5 prime to 3 prime, and this is here also 5 prime to 3 prime only. So now these two are called anti-parallel to each other. One is coming from 5 prime to 3 prime and this also coming from 5 prime to 3 prime. But both are moving in opposite directions. Both are moving. So these two are parallel strands, but both parallel strands are passing in opposite direction. In opposite direction, so that this is called anti-parallel. This is one nucleotide. And nucleotide contains base and phosphate groups. The phosphate groups are bind to nucleotide with the help of phosphodiester bond. So that in a nucleotide strand, in a polynucleotide strand, the two nucleotides are linked together with phosphodiester bond in a nucleotide. The base is binded to ribose sugar with the help of glycosidic bond, and in between two polynucleotide strands, two bases are binded together with hydrogen bonds. So this is hydrogen bond and this is glycosidic bond. This is glycosidic bond. Base is bind to ribose nucleotide, ribonucleotide. So with the help of glycosidic bond, and the two nucleotides are combined together with phosphodiester bond. This is one phosphodiester bond and this is one phosphodiester bond. Totally phosphodiester bond we can see. And in between two bases hydrogen bonds are present. These are hydrogen bonds. And each two strands of polynucleotide strands are passing in or anti-parallel direction. So both are passing 5' to 3' direction. In a polynucleotide strand, one nucleotide is connected to its successive nucleotide. One nucleotide connects to its successive nucleotide with 3' end. So that here 3' phosphate group is very, very important. If 3' OH group is very, very important to continue polymerization. For example, in a polynucleotide strand, 3' OH group is removed. If 3' OH group is removed in a polynucleotide strand, there will be no further polymerization process. For the polymerization process, 3' OH group is very, very important because always the polynucleotide strand synthesis takes place in 5' to 3' direction. So that 3' OH group is very, very essential for polymerization process. If 3' OH group is removed in a nucleotide, then the polymerization will be stopped in that place. The characters of polynucleotide strand. Polynucleotide strand contains two strands, and the two strands, sugar and phosphate are present here, sugar and phosphate. The sugar phosphate bond is backbone to polynucleotide strand. Sugar and phosphate groups are present over the stacks, and one nucleotide stacks over another nucleotide, and this, as the hydrogen bonds present in the center of the polynucleotide strands, this phosphate and sugar bond only gives stability, to or this is the backbone to polynucleotide strand. So sugar and phosphate group is the backbone for polynucleotide strand, and the distance between two strands of polynucleotide strand is 20 angstroms. And polynucleotide strand is anti-parallel to each other because one strand moves in opposite 5' to 3' and another strand moves in 3' to 5'. So in this way, so both are parallelly they will present, but they go in opposite directions. The direction of the synthesis of polynucleotide strand always takes place from 5' to 3' end or 5' to 3' end. But in one strand passes in this direction 5' to 3' and another one more strand passes through this 5' to 3', 5' to 3'. In this way, so the both strands in a polynucleotide chain, both strands are anti-parallel to each other, So this is a right-hand helix DNA, are also known as bDNA. This is right-hand helix DNA forms a full turn. A full turn contains 10 base pairs. A full turn contains 10 base pairs. So there are totally 4 types of DNAs are very common. The 4 types of DNAs are, so first one is aDNA, bDNA and cDNA and zDNA. These 3 are right-handed DNAs and zDNA only left-handed DNA. And aDNA, bDNA, all these 3 are right-handed DNAs. Because the helix turns towards the right side. aDNA, bDNA and cDNA, these are right-handed DNA. A full turn contains totally 11 base pairs in aDNA, and bDNA, a full turn contains 10 base pairs, and cDNA, a full turn contains only 9, and in the zDNA, a full turn contains 12. We are discussing about the characters of bDNA, so that we have discussed in between in a full turn 10 base pairs are present. Once again let us see. There are three types of DNAs are there. There are three types of DNAs are there. The first one is A DNA, and second one is B DNA, and third one is C, and the third one is Z. A DNA contains one full turn 11 base pairs. B DNA contains 10 base pairs. C DNA contains 9 base pairs in one full turn. Z DNA contains 12 base pairs. But the first three are right-handed DNAs. The first three are right-handed DNAs, and Z DNA is only left-hand DNA. The left-hand DNA, Z DNA contains 12 base pairs in one full turn. So now we are discussing the characters of B DNA. So B DNA also right-hand helix. In the right-hand side, the helix takes place. So then this is one full turn. A full turn contains, from here to here, a full turn contains 3.4 nanometers. The length of a full turn is 3.4 nanometers or 34 angstroms. 3.49 nanometers or 34 angstroms, because 1 nanometer equals to 10 angstroms. 1 nanometer equals to 10 angstroms, so that 3.4 nanometers, one full turn length is 3.4 nanometers or 34 angstroms. So that in the one full turn, totally 10 base pairs are present, so that each base pair, the distance between each base pair is only 0.34 nanometers, 0.34 nanometers or 3.4 angstroms. The distance between two base pairs, is 3.34 nanometers or 0.34 nanometers or 3.4 angstroms. And the angle between two full turns is 36 degrees. The angle between two full turns is 36 degrees. And that distance between two polynucleotide strands is 20 angstroms. The distance between two polynucleotide strands is 20 angstroms. And what is the backbone for polynucleotide strand? Sugar and phosphate group is the backbone for polynucleotide strand.